It's my great honor to introduce our first speaker, Daniel, who is a MD PhD student at Harvard Medical School. So Daniel obtained his bachelor degree from Yale University from molecular biophysics as well as biochemistry. And then he became a MD PhD student at Harvard Medical School with Diana and Matthias, as well as Christopher Beno's lab, where he is focusing on the thymic mechanisms of T-cell tolerance. So he finished his PhD and is currently performing his MD at the Harvard University. And during his PhD, he actually has been involved in multiple events, such as medical evaluator, the senior clinician, and he has also been as a teaching assistant for computational biology for Harvard University. He has also presented in several conferences about his thesis work. So with that, I will hand the floor to Daniel to tell us about his thesis work.
Yeah, awesome. Thank you, Sky. Can everyone hear me all right? No protest.
Yeah.
I'll go for it. Yeah, so thank you, Sky. I'm very excited to share with you all today some of my thesis work studying how gene control happens in the thymic epithelium and sort of thinking about how that relates to how we think about gene control elsewhere. So I thought I would start, I know this is not necessarily an audience of immunologists, so I thought I would just start with some high-level background about how the immune system develops and what the particular role of the thymus is. So I'm very interested in T-cell development in particular, and T-cell development largely happens in the thymus. And what basically happens is we develop this large pool of randomly developed T-cells that can basically represent an incredible, or sort of recognize an incredible diversity of different antigens, so protein antigens. And so we get this amazing diversity, and that allows our immune systems to do all the amazing things that they can do. But there is sort of a caveat with this, which is that by random chance, some of our T cells will also recognize our own protein antigens. So these are basically autoreactive T cells. And if they're allowed to go out into the periphery, they can go out and attack our own tissues and cause autoimmunity. And so in the thymus, we actually have a mechanism known as negative selection, whereby we can actually identify these autoreactive T cells and remove them from the repertoire before they have a chance to cause autoimmunity. And kind of zeroing in a little bit on how this process works at a sort of more cellular molecular scale, we have this very specialized cell type called the medullary thymic epithelial cell, or MTEC. And what these MTECs do is they sort of preview the peripheral self to T cells. And what I mean by that is they ectopically express thousands of so-called peripheral tissue antigens, or PTAs. So these are basically antigens which are normally expressed very specifically in particular tissues, such as insulin in the pancreas, mucins in the gut and the lung, or myelin components in the central nervous system. MTECs somehow turn these on within the thymus, so the T cells kind of get a chance to preview what the rest of the body looks like. And any autoreactive T cells can be identified and removed in the thymus before they can cause damage. Now, about 20 years ago, my lab was actually very important in showing that this single transcriptional regulator called autoimmune regulator, or AIR, is very important for the expression of these PTAs. So if you delete AIR, you downregulate expression of thousands of these PTAs and MTECs, and AIR turns out to be very important for tolerance because actually both mice and humans with mutations in AIR go on to develop multi-organ autoimmunity. Now, AIR is kind of interesting from a gene regulation perspective because it doesn't really seem to follow the classical rules of how we think about transcription being regulated. So you don't have these big blocks of like co-regulated genes that you might think of being controlled by a classic transcription factor. AIR doesn't really have a sequence specificity. And instead, it's been proposed that AIR basically acts stochastically, probabilistically, or quasi-randomly to turn on these different PTAs within MTEC. So it's been sort of a mystery of how AIR and this PTA expression works. So at the start of my PhD, I was actually very interested in whether we could better understand this process of PTA expression using advances in genomics, and especially with recent advances in single-cell genomics. And so one of the early experiments that I did was performing single-cell attack sequencing of MTECs from either air wild type or air knockout mice, and kind of allowing us to look at the chromatin accessibility in individual MTECs. And I should say that for this first part of the talk, a lot of this work was published in this article last year. So starting first with air-dependent differences, in this sort of main body of cells here in the center of the UMAP plot, you can see that in the air wild type condition, most of these cells localize to this single air positive cluster, whereas in the air knockout condition, you basically totally lose those air positive cells and the cells shift over to the right of the UMAP space, suggesting that air has a very substantial impact on chromatin accessibility in MTECs. Now, I have to admit that this is actually a little bit of a red herring for this talk, because we actually ended up focusing much more deeply on a second aspect of the data, which we hadn't really expected, which was this surprising heterogeneity in these cells, which had been known as post-air MTECs. And so these were basically cells which had previously upregulated and then downregulated air. They had been described in 2013 and 2014 by a couple of groups, but really not much was known about their development, their function, their roles in tolerance, not much at all. But what was interesting to us is when we started to dig into these post-air cells, we realized that we could discriminate these different clusters from each other and from other MTECs by their high accessibility of specific transcription factor motifs within their accessible chromatin. So for example, in these brown and blue clusters here on the left, there was high accessibility of FOXA and FOXJ family motifs. In this gray cluster here in the center, there was high accessibility of grainy head-like or GRHL family motifs. And in this sort of tan cluster over here, high accessibility of hepatocyte nuclear factor 4 or HNF4 family motifs. Now, if you're anything like me, you're probably thinking, okay, that's kind of interesting, but I haven't necessarily heard of grainy head-like or hepatocyte nuclear factor 4 before. I hadn't either. But I'll just sort of skip to the punchline and tell you that these turn out to be really key lineage-defining transcription factors for a variety of different peripheral tissues and cell types. So, for example, grainy head-like, which defined this gray cluster here in the center, turns out to be very important for skin development. And so mice that lack one of these grainy head-like homologs actually never form an impermeable skin barrier, and this becomes a lethal defect for them. Similarly, HNF4 is very important for the development of the gut and liver epithelium, and without different combinations of HNF4 homologs, you get very severe liver gut dysfunction. So we saw these small subsets of MTECs that were marked by these lineage-defining transcription factors, and for us, that gave rise to this hypothesis that we know air is very important for this process of PTA expression in the thymus, but what if alongside air, we have this sort of second process, which hadn't really been well appreciated, where we actually had lineage-defining transcription factors driving the same gene programs that they drive in the periphery, but now in the thymus for the purposes of T-cell tolerance. And so that was sort of the hypothesis that we then set out to pursue. And we started by really focusing in now on these post-air cells and just performing single-cell RNA-seq of that compartment to kind of capture the full diversity there. And when we did this, we could see this really amazing diversity of different peripheral tissue types and cell types represented within the thymus. Collectively, we call these mimetic cells because they're sort of mimicking the periphery. So we could see mimetic cells that we expected from the single-cell attack sequencing, such as MTECs that look like skin or MTECs that look like gut and liver epithelium. With this higher resolution look, we could also start to see new subtypes, such as a subtype that look like skeletal muscle, or a subtype that look like ionocytes, which are the cystic fibrosis channel expressing cells in the lung. So really this kind of amazing diversity of different cell types within the thymus. And we could identify all of these different clusters of cells because if we took gene signatures which were derived from the bona fide cell types of the real ionocytes or real enterocytes, and then overlaid these gene signatures onto our MTEC data, we could see very focused expression of these gene signatures in these particular clusters of different MTECs. And if you'll remember what I told you about how air-dependent PTA expression was thought to work in this sort of stochastic, quasi-random process, this is clearly something very different where we have very biologically logical and coherent gene expression within MTECs. And one other point, which I think is important, is that for every mimetic cell subtype, we could very readily identify a transcription factor or a set of lineage-defining transcription factors, which correlated with the expression of these lineage-specific programs and, I guess, putatively controlled their expression. So we were pretty surprised to see this, especially kind of given our preconceived notions about how this process had worked going in. But once we started to kind of wrap our heads around it, we then wanted to hone in on mechanistically how this was possible. And so we started to ask what the role of these lineage-defining transcription factors was in driving the differentiation function of these mimetic cells. So the first thing that we did was we profiled their chromatin landscapes. So we started by looking at AIR ChIP-seq, which had been generated by a previous postdoc in the lab, Kushagra Bansal, as well as H3K27 acetylation, marking active enhancers and promoters, at a variety of different open chromatin regions corresponding to different mimetic cell subtypes or other MTEC subtypes. And so you can see AIR and K27 acetylation are pretty evenly distributed across these different mimetic cells subtypes, so not too much of a difference there. However, when we looked at lineage-defining transcription factors, we saw something very different. So for example, POU2F3, which, as I think Skye knows well, is the key lineage-defining transcription factor for tuft cells, POU2F3 really only bound to the tuft mimetic cell OCRs, but not to other mimetic cell OCRs. Similarly, HNF4 alpha and HNF4 gamma, which are the key lineage-defining transcription factors in enterocytes and hepatocytes, really only bound to these enterohepatocyte mimetic cell OCRs, but not others. A little bit at the microfold OCRs, which I'm happy to discuss if people are interested. And similarly, grainy head-like 1, again, the skin transcription factor really only bound to these keratinocyte-specific OCRs, but not other OCRs. And there's very little background in this assay. So from this, we concluded that lineage-defining transcription factors seem to bind chromatin specifically within mimetic cells. Next, we wanted to understand what these transcription factors were actually doing to the chromatin of mimetic cells. And so to do this, we generated mice where we could conditionally ablate both HNF4 homologs, HNF4 alpha and HNF4 gamma, within thymic epithelial cells. So those are HNF4 delta tech mice, and compare them to control mice. And then we profiled a variety of different histone marks and chromatin-associated factors to kind of get a sense of what HNF4 might be doing to mimetic cell chromatin. And so from this kind of high-level view, I think you can appreciate that, one, we see our different marks distributed kind of where we expect them, so the data look good. And two, when we compare between genotypes, we don't see gross differences in the distribution of these different marks in the presence or absence of HNF4, suggesting HNF4 is not sort of like grossly remodeling MTEC chromatin. However, then we started to look very specifically at these enterohepatospecific open chromatin regions, which were the ones where we would kind of expect HNF4 to be acting based on our cut and tag data. And when we did this, indeed, we could see a very substantial increase in K27 acetylation in the presence of HNF4. Suggesting that HNF4 is activating enhancers and or promoters. To kind of parse out which one it was, we also looked at K4 monomethylation, K4 trimethylation. Here, again, we saw K4 monomethylation was increased in the presence of HNF4, albeit not quite to the same extent as K27 acetylation. But K4 trimethylation really wasn't affected by HNF4, suggesting that HNF4 is primarily activating enhancers to mediate its effect. Its effects. Consistent with this, when we looked at K36 trimethylation marking active gene bodies, we also really didn't see much of a difference in the presence of HNF4. We looked at a few other factors, so K27 trimethylation marking polycomb-mediated repression. There really was no significant differences in the presence or absence of HNF4 there, so we don't think it's affecting polycomb. And then interestingly, CTCF, which of course is a very important 3D structural factor, was focally enriched right at these enterohepatospecific OCRs in the presence of HNF4. So we do think that HNF4 is probably either directly or indirectly redistributing CTCF in MTECs as well, and probably having some influence on the 3D structure of mimetic cells. And again, there's very little background in these assays suggesting that these are all specific effects. Okay, and then the last point what is the more sort of like cellular and immunological phenotype of these HNF4 knockout mice? Are these lineage-defining transcription factors actually required for the mimetic cells? So we figured out how to gate for these enterohepato-MTECs by flow cytometry. They're about 1% of total MTECs. And indeed, in the HNF4 delta tech condition, you basically totally ablate this compartment cellularly. And then when you look at MTECs more broadly, and remember, they need to express many, many transcripts to tolerize your T cells, we see a very focal downregulation of about 150 transcripts, most of which are these gut and liver associated genes. So apolipoproteins, antimicrobial peptides, classic gut markers like vilin, all downregulated in the absence of HNF4. So, oh, and I'll just mention, I don't have time to go into it all here, but at this point, we have similar results for a variety of other lineage-defining transcription factors and other mimetic cells. We and others have published results. So we think this is likely to be a general phenomenon, these different mimetic cells requiring these different lineage-defining TFs. So I'll just kind of wrap up here with a model of how we think this is working, how we think gene control is happening in the thymic epithelium. So on one hand, we have our sort of canonical model where we have sort of quasi-random induction of PTAs by air within these air-expressing MTECs. And it's not that we think that this model is wrong. Indeed, I showed you at the beginning that there are very strong air-dependent differences in MTEC chromatin state. But in addition to this, we have this sort of second aspect, which I don't think has been well appreciated until now, where we actually have very coordinated PTA expression driven by these lineage-defining transcription factors in these sort of diverse mimetic cells. And again, all of this is for the purpose of taking these autoreactive thymocytes or T cells, which are naturally generated during the development of the immune system, and either removing them from the immune repertoire or diverting them to a tolerogenic fate. So I'll just acknowledge all the people that have made this work possible, especially my primary mentor, Diane Mathis, who has been a really great mentor and a great person to work with. Chong Zhuo helped me with some of the HNF4 experiments, and then Michael Verzi very generously provided us with the HNF4 conditional mice. So very grateful to him, and I'd be happy to take any questions.
And thanks, Daniel, for the great presentation. So if you have any question, you can either type it in the chat box or you can raise your hand. So we already have one question, so I'm going to unmute you so you can speak.
Hi, my name is Katerina Gurova, and it's very, very interesting. It's interesting biological and interesting from transcriptional standpoint. I have two quick questions. First, did you look at the cells with a similar transcriptional profile, whether they are located close to each other in thymus or they are completely kind of randomly distributed? And my immediate idea whether thymus looks like a teratoma in this regard.
Yeah.
And second question is, it is clear from your studies that these lineage regulating transcriptional factors defines transcriptional program. Did you look in the role of air in the control of expression of transcriptional factors per se?
Yeah, those are both great questions. So for the first one, it seems to vary a little bit by different types. So we see some mimetic cells kind of cluster with other things that you might imagine to be sort of lineage related. So for example, we see these cystic structures in the thymic epithelium that sort of look like airway. So you have ciliated cells that are lined up next to mucus producing cells, which are lined up next to like ionocyte-like cells. So in that respect, there does seem to be some sort of clustering, if you will, but for other cell types like I guess the microfold in the gut, which we sort of for different reasons think are related, there doesn't seem to be as much clustering, although we haven't done like a rigorous some sort of simulation-based testing to see if it's like non-randomly distributed. So I think it's both sort of flavors on that point. And then with regard to whether air regulates the lineage-defining transcription factors, I think the jury is still out a little bit. We do have some data. So we did single-cell RNA sequencing of our mimetic cells in the presence or absence of air. And some subsets are very dependent on air for their differentiation. So like the ciliated MTECs almost totally disappear in the absence of air, whereas others, such as muscle MTECs, really don't seem to depend on air at all. But whether there's a molecular relationship between air and the lineage-defining transcription factors, I think we still need to do more work to kind of tease that apart.
Thank you.
Yeah.
So I have a question. So you need these epithelial cells to educate the T cell. Then I would be shocked that there are not more cells are being expressed, like in here, for example, like neuroendocrine cells, goblet cells. Like what happened to them? Like why are there no expression program for these cells to educate the T cells?
Yeah, it's a great question. So I mean, one thing is that we do see actually a pretty wide diversity of cells. So we do see like a neuroendocrine subtype. As we kind of accumulate more cells, we kind of can pull out more of these different subtypes. But the other point that I would say is that I kind of think of these as like cell archetypes, as opposed to like specific cell types like you would see in the periphery. So you know, like I've labeled them here by their different sort of organotypic characteristics, but really I think they're transcription factor driven entities. And so like HNF4 drives a program that's related to gut and liver. You kind of get this like hybrid phenotype that really is a function of the transcripts that they're or the antigens that they're expressing, and not necessarily the organ where they would be situated. So I think you're kind of tolerizing against transcriptional programs as opposed to specific cell types, if that makes sense.
Yeah, so there's one more question. I'm going to unmute.
Hi, very nice talk. I'm Matt Lavin from Greece. So I have a question regarding your HNF4 DTEC analysis.
Mm-hmm.
So you say that these guys that are downregulated in the gut, they are probably markers of your lineage that would be liver, yeah, in your knockout mice.
Yeah, like many of the transcription or the transcripts that are downregulated in the absence of HNF4 are sort of
exactly.
Yeah.
So your cells practically they resemble liver cells, although they are in the thymus.
Yeah,
based,
I mean, I guess
based on your single cell analysis, I don't think you have any, let's say, pathology or whatever slide to prove it. It's interesting though. So my question is more philosophical. I mean, okay, so you would have maybe some functional cells in the thymus that could do some, let's say, function of the liver or whatever other organs you say, and you found. But what is the proportion of these mimetic cells in the thymus?
Yeah, so I mean, we do see varying degrees of like phenotypic overlap, I guess, to that point. So like the ciliated cells really do have like cilia coming off the top of them, and you can see those sorts of like macro structures. So and sometimes it can be quite a dramatic phenotypic conversion. Although when we look transcriptionally, comparing to the bona fide peripheral epithelium, they seem to be more similar still to thymic epithelial cells than the bona fide peripheral epithelium. So they adopt some aspects, but kind of retain the identity.
Okay.
As to the relative mix of different cell types, I think we still don't really understand what's determining why some are way more abundant than others. So like in the mouse, tuft MTECs are like very abundant for some reason.
Okay.
It's not clear like if that would correlate with the degree of tolerance that you would need against them, or if they have some other role. But yeah, I don't think we totally know.
And are they compartmentalized in your tissue, or there's like a pocket with those cells, or they are just hanging around everywhere so that the cells have a chance to scan them?
Yeah, most of them are quite diffusely distributed throughout the thymus. A few. Sorry?
It's not really structured per organ.
Yeah, it's not like you have like one lobe of the thymus that's like just gut. But I mean, again, for some subtypes like the respiratory epithelium, those seem to be a little bit more kind of focally concentrated.
Yeah, maybe it could be worth trying to do some Merfish analysis on those guys.
Yeah. So we are trying to do some like high dimensional imaging now to
Okay.
capture all the different subtypes with relation to one another.
Beautiful. Thanks.
Thank you.
So Daniel, you have more questions at the Q&A box, and because of the time limit, so probably we can't answer them
Okay.
by
maybe I
person.
can just
So yeah,
type
so you can, if you can just type the questions,
Great.
like your answers, I would really appreciate.